Hi everyone, my name is Katie Griffin and I'm a graduate student at East Carolina University. Today, I'm going to look at what the impact is of a vegan and vegetarian diet on gut microbiota. The objectives of this presentation are to discuss background on diet, the microbiome, and gut health, examine what, if any, impacts a vegan or vegetarian diet has on the gut microbiota compared to an omnivorous diet, and lastly, discuss the conclusions, implications of any findings, and next steps. So how prevalent is vegetarianism? Vegetarian diets have become more popular over the years, but we've seen the most growth in the United States in the last 3 years. According to the Statista Global Consumer Survey, in 2021, 5.1% of Americans responded that they followed a vegetarian diet. This is up from 2.9% in 2018. About 2% of the U.S. population reported that they followed a vegan diet. Based on the current U.S. Census, that means about 17 million Americans eat a vegetarian diet and just under 7 million Americans eat a vegan diet. You'll notice that in other countries such as Great Britain, Germany, and South Korea, vegetarianism also has grown over the last few years. But in other countries such as India and China, vegetarianism has fallen quite substantially as a more westernized diet has become more popular. As a recap, there are different kinds of vegetarian diets. The overarching vegetarian diet does not include meat or poultry, but there are a few variations. Pescatarians consume fish, ovo-vegetarians consume eggs, lacto-vegetarians consume dairy products, and then there are vegans, which eat no meat, poultry, fish, eggs, dairy, or other animal products such as gelatin or honey. So now let's talk about the gut microbiome and microbiota. The gut microbiome is made up of trillions of microorganisms, also known as microbiota or microbes, that live in the intestinal tract, some of which can be helpful or potentially harmful. These not only include bacteria, but also fungi, parasites, and viruses. Intestinal bacteria play a critical role in protecting against pathogens and maintaining metabolic homeostasis. DNA originally determines a person's makeup of microbiota. We are first exposed to microorganisms as an infant, during delivery in the birth canal, and then through the mother's breast milk or formula. As we get older, environmental exposures and diet can change our microbiome to either be beneficial to health or to put us at greater risk for disease. Gut microbiota are involved in a number of critical bodily functions, such as regulating immune function, protecting against pathogens, and collecting energy from digested food and maintaining metabolic homeostasis. So as you can imagine, any changes to the microbiota composition can affect all of these functions. This image to the right represents the role of the gut microbiota in health and disease and gives some good examples of inputs and outputs. So you've got some negative outputs over here, such as insulin resistance, gut inflammation, increased cardiovascular disease risk. These are all negative impacts of having dysbiosis and having more of the quote-unquote negative bacteria. And then you've also got positive outputs, and this is from gut bacterial homeostasis, such as increased short-chain fatty acid production, improved lipid metabolism, low gut inflammation, and insulin sensitivity. An increasing body of evidence suggests that maintaining a healthy gut microbiome has a profound impact on our health. Dysbiosis, which is an imbalance of gut bacteria composition, has been associated with the development of many inflammatory conditions and diseases, such as inflammatory bowel disease, psoriatic arthritis, diabetes, atopic eczema, and celiac disease, to name a few. You can see from the figure on the right a number of diseases and conditions that can be implicated in part by an imbalance of gut microbiota. In addition to a person's genes, environment, and medication use, diet has one of the largest influences in determining what kinds of microbiota live in the gut. On average, 60 tons of food and environmental microorganisms pass through the human GI tract throughout a person's lifetime. This imposes a huge threat to gut integrity. The close relationship between diet, the gut microbiome, and health suggests that we may be able to improve our health by modifying our diet. So here we have a really nice visual of this. So we've got the dietary intake, specifically more so the nutrients, so protein, fat, carbs. We ingest that. There's an alteration in gut bacteria. So for example, changes in the bifidobacteria, lactobacilli, Akkermansia. You've got the biologic effects that causes. So, for example, an alteration in host metabolism, immune system production of pro and anti-inflammatory metabolites, and it eventually leads to host disease. So let's start with an overall list of some bacteria in the gut that's commonly affected by diet. The green are species of bacteria that have been found to be generally beneficial, and the red are bacteria found to be generally pathogenic or harmful. To call out a few that we'll discuss in this presentation, we'll hear about the bifidobacterium species, which is involved with short-chain fatty acid production, and it improves gut mucosal barrier. Lactobacillus species, this is one we've probably all heard about from probiotics. It's also involved in short-chain fatty acid production, as well as anti-inflammatory and anti-cancer activities, and it's known to reduce effects of inflammatory bowel disease. The Bacteroides species activate CD4 and T cells and are found to be more abundant in inflammatory bowel disease. And then we've got the Allostipes, Bilophila, and Clostridium species, and they've all been found to be abundant in pro-inflammatory processes and infections. Just continuing on with this chart, we've got the Roseburia and the Eubacterium species. They're also involved in short-chain fatty acid production and are found to be low in inflammatory bowel disease. The Faeculum prosnitzi is also involved in short-chain fatty acid production and has anti-inflammatory effects. It plays a protective role in metabolic disease, and low levels of this are associated with intestinal disorders, inflammation, obesity, and type 2 diabetes. And then we've all heard about E. coli, H. pylori, Streptococcus. These bacteria are typically involved in inflammation of the stomach and intestine. They're all pretty pathogenic. Before we jump into some studies, I want to show sort of an overall hierarchy of the gut microbial composition. So in this blue box, you'll see examples of bacteria belonging to the Firmicutes and the Bacteroidetes phyla, which represents approximately 90% of the gut microbiota. We'll see that these studies differ and oftentimes have contradictory results, and that's really just based on their different methodologies and the taxonomic level of bacteria studied. So for example, we've got the Bacteroidetes, it's the overarching phylum. In some studies, it was found in abundance in a low-fat vegan diet, but then you've got the Bacteroides species, which is a more specific species that falls within the Bacteroidetes phylum. And in some studies, that was found to be in more animal-based westernized diet. So it's not comparing apples to apples. So the first study that we're going to look at is a study by De Filippo et al., which investigated the impact of diet in shaping the gut microbiota. So in this study, researchers compared fecal microbiota of European children to children in a rural African village. The African children ate a diet rich in whole plant foods, predominantly vegetarian, with a very low intake of animal protein. Their diet mainly consisted of cereals, such as millet grain and sorghum, legumes, and vegetables. And the European children, which were from a more populated area in Florence, Italy, ate a typical Western diet, high in animal and protein and fat, starch and sugar, and low in fiber. The results of the study reported that African children had higher gut microbial richness and biodiversity than the European children. So a decreased gut microbial diversity is a proposed indicator of an unhealthy microbiome and has been linked to different chronic conditions such as obesity and type 2 diabetes. The microbiome of the African children was dominated by the Prevotella enterotype. What's interesting is that the Prevotella and the Zylantobacter enterotypes were found exclusively in the African children's microbiota in this study. This is shown in the green shading on the left. So we've got these two. We've got the Prevotella and the Zylantobacter. Those were, again, primarily prevalent in the microbiome of the African children. This could be due to a higher fiber intake from the plant polysaccharides in the diet of these children. Conversely, the microbiome of the European children was dominated by the Bacteroides and the Faeculobacterium enterotypes. So we've got these two right here. So again, just pointing out the difference in the bacteria between these two groups, the two cohorts of children that had different diets. One, a more vegetarian diet, and one, a more animal-based diet. In terms of metabolic activity, the African children had almost 3 times the concentration of short-chain fatty acids compared to the European children, which is outlined here. Additionally, the European children had higher relative abundance of potentially pathogenic enterobacteria, including E. coli, Salmonella, Shigella, and Klebsiella. The researchers concluded that the results suggest that diet has a dominant role over the other possible variables, such as ethnicity, sanitation, hygiene, geography, and climate, in shaping the gut microbiota. So this is a second study where researchers investigated the association of dietary and environmental variables with gut microbiota. The first phase was a cross-sectional analysis of 98 healthy participants, where researchers collected diet information using 2 questionnaires that probed recent diet as well as long-term diet via food frequency questionnaires. DNA samples and stool samples were obtained at the beginning and the end of the study. And then there was a second phase where 10 participants were sequestered in a hospital environment in a controlled feeding study to compare a high-fat, low-fiber diet and a low-fat, high-fiber diet. Stool samples were collected and DNA samples were analyzed, and a rectal biopsy samples were taken on days 1 and 10 of this second phase. Researchers in this study built on results of previous research on enterotype association with different dietary profiles. And they determined that most people have prominence of the Prevotella and the Bacteroides enterotype in their gut. The Prevotella enterotype was associated with a high intake of carbohydrates and simple sugars, which is typical of a carbohydrate-based vegetarian diet. And that was evidenced by the 11 vegetarians and 1 vegan in the study, which showed enrichment of the Prevotella enterotype. On the other hand, the Bacteroides species is highly associated with animal protein and saturated fat intake in this study, which suggests that a more Western diet characterizes this enterotype. In this chart on the right, you'll see the Bacteroides on the left and the Prevotella on the right. The blue means very little abundance, and the red means a higher abundance in the gut. So you'll see that when looking at more of the carbohydrate sources, the Bacteroides is blue, meaning very little prevalence in the gut, and the Prevotella is red, meaning a higher abundance in the gut. The opposite can be seen for animal sources and fats. So Bacteroides is dark red, meaning very high abundance, and Prevotella is blue, meaning less abundance. So as we saw in the last slide, associations found in phase 1 of the study parallel the study we just talked about by De Filippo et al., which compared the diet of the European children to that of children in a rural African village. For the phase 2 of the study, changes in the microbiome composition were detectable within 24 hours of initiating the controlled feeding. However, researchers didn't notice any meaningful or stable change in the microbiome after 10 days of switching half the patients to the low-fat, high-fiber diet, which is evidenced here. So we see a change on day 1, but then we really don't see any meaningful change after that. Researchers concluded that based on their data, long-term diet is strongly associated with enterotype dominance, particularly protein and animal fat with Bacteroides versus carbohydrate intake with the Prevotella. While changes in microbiome composition were detectable within 24 hours of initiating the controlled feeding, they were modest and they were not sufficient to switch the participants between the 2 enterotypes long-term. These researchers also emphasized the importance of determining whether individuals with the Bacteroides species have a higher incidence of diseases associated with the more Western diet, and whether long-term dietary interventions, such as changing to a plant-based diet, can switch individuals to have the Prevotella enterotype. Here we have a summary table of the effects of non-digestible carbohydrates on gut microbiota. The authors took a number of the studies that we just discussed and extracted nutrient-specific findings from vegetarian and omnivore diets. So as we know, a vegan vegetarian diet is known to be rich in fiber, prebiotics, and resistant starches. So looking at the intake of the fiber and prebiotics, which comes exclusively from plants, resulted in increased bacterial abundance and gene richness, which has been a proposed hallmark of gut health and healthy microbiome. It yielded an increase in commensal or positive bacteria, such as the lactobacilli, bifidobacteria, and a decrease in pathogenic Clostridia. And here you see, again, conflicting results on the studies, and I think a lot of that comes down to just the difference in how the studies were performed and at what taxonomic level they were reporting. So then if we take a look at the resistant starch, So foods such as oats, rice, legumes, and potatoes, higher carbohydrate intake, also yielded an increase in the bacterial abundance and gene richness, and also resulted in an increase of the lactobacilli, bifidobacteria, the roseburia, the eubacteria, and the ruminococcus. From this same review article, we have a summary regarding how both the amount of fat and type of fat impact the microbiota. Now, looking specifically at the type of fat, saturated fats, while there are some plant-based foods that are naturally higher in saturated fats, they're mainly found in animal foods. An intake high in saturated fats led to an abundance of the Bacteroides, Bilophila, and Faecalibacterium prausnitzii. The Faecalibacterium prausnitzii has actually been found to be a beneficial bacteria when looking at the short-chain fatty acid production. So again, I think this is a good example of differences in data depending on what exactly is being studied. With a higher unsaturated fat intake, which are predominantly found in plant foods such as avocados, seeds, a variety of nuts and vegetables. The unsaturated fat intake was found to increase the lactobacillus and the streptococcus species, as well as the bifidobacteria and the Akkermansia mucinophila, all of which have been shown to have beneficial effects. So in figure 2 on the left, we have plant protein at the top and animal protein at the bottom. With an intake of plant protein, which is predominantly found in vegan and vegetarian diets, researchers generally found an increase in the bifidobacterium and the lactobacillus. Both are beneficial or commensal bacteria, and a decrease in the Bacteroides and Clostridium perfringens. We also see an increase of the short-chain fatty acids, an increase in improvement in gut barrier, and a decrease in inflammation. So short-chain fatty acids such as butyrate provide all kinds of benefits. They help our body absorb essential minerals, increase satiety, decrease inflammation. So an increase in short-chain fatty acids is a good sign. With an intake of animal protein found primarily in the omnivorous diets, researchers generally found the opposite of a plant protein. So increase in Bacteroides, allostipes, Bilophila, ruminococcus, and a decrease in the beneficial bifidobacterium. We also see an increase in TMAO, which increases the risk of cardiovascular disease, as well as a decrease in the short-chain fatty acids, which has been linked to inflammatory bowel disease. So the relationship between diet and the intestinal microbial profile appears to follow a continuum, with vegans displaying a gut microbiota most distinct from that of omnivores, but not always significantly different from that of vegetarians. Studies that have involved intake of specific dietary components like protein, fats, digestible and non-digestible carbohydrates, probiotics, and polyphenols demonstrate how certain bacteria tend to respond either by having an abundance or a reduced abundance in the human gut. So how the microbiota respond to the dietary component itself versus the source of the food. How our gut responds, again, by having a production or lack of these bacteria, can have positive or negative impact on our body and overall health. In conclusion, findings from current literature suggest that fundamental dietary choices, such as vegetarian or omnivore diets, do influence the microbiota. For example, consuming a plant-based, high-fiber diet increases exposure to a variety of environmental microbes and can increase beneficial bacteria in the gut. And benefits to having more of the positive bacteria, such as lactobacillus, can ward off inflammation, decreasing the odds of certain GI disorders and diseases. The conclusion of these studies discussed illustrates the importance of role of gut microbiota in human health and possibly suggests that manipulating them may influence disease activity, but more research is needed. So overall, further studies with careful experimental design are needed to provide more insight into the differential effects of vegan and vegetarian diets on the gut microbiome. While the current literature is helpful, researchers are still really in the early stages of understanding the microbiome's role in human health. So again, further research is needed, but this is all very promising. Thank you.